Greetings to all. My name is Vira, and I'm here with my fellow Vira. We are both statistics students and are from Intro to Bioinformatics class. Today, we would like to present our review on a paper written in 2023, which is titled Soil Microbial Community Compositions and Tolerance to Contaminant in an Urban Brownfield Site. The problem this paper decided to focus on is severe environmental pollutants, for example, heavy metals, PAH, and benzenes, which those reduce overall microbial diversities. Through this, it favored the growth of resilience of microbial with the mechanisms to transform, neutralize, accumulate, or exclude pollutants. However, this paper states that about 50% of urban land in the USA are contaminated brownfields, which they are toxic, compact, nutrient-poor soils harm ecosystems, where restoration is needed. However, it needs the understanding of biodiversity and soil microbes. This paper suggested bioremediation using native microbes. It is safe and sustainable. The research gaps for this paper is that there are very few studies discussing soil microbiome across varying depth of soils. Hence, the goal of this paper is to characterize the impact of long-term environmental contamination across multiple depths of soil. And the second one is to identify specific microbial taxonomic groups.
The study used environmental DNA metabarcoding with three genetic markers, 16S rRNA for bacteria, 18S rRNA for eukaryotes, and ITS for fungi. A total of 55 soil samples were collected from 5 depths. The researchers analyzed microbial diversity and community composition using linear mixed models, Permanova, principal coordinate analysis, canonical correspondence analysis, and linear discriminant analysis effect size. These methods help identify the relationship between microbial communities and soil contaminants. The response variables were microbial community composition, alpha diversity, and beta diversity, while the predictor were soil depth and contaminant concentrations such as arsenic, chromium, cobalt, lead, petroleum hydrocarbon, and polycyclic aromatic hydrocarbon.
There are three main topics for the main finding for this paper. The first one is contaminant-driven diversity, where they found that 18 sRNA community richness was positively correlated with lead concentrations, and the second one is fungal richness was positively correlated with cobalt concentrations. And for the second main finding is for the depth matters of the soil, which they found that microbial communities vary significantly across the sample depth that they took, which accounted for a notable portion of the microbiome variations. The third one is for enriched taxa for bioremediations, which they successfully identified the heavy metal tolerant taxa that were significantly enriched in contaminated surface soils, which the taxa are listed as follows. The model created in the studies are able to explain the conditions of microbiome in soil, which soil depth explain about 6 to 9% of microbiome variations. Heavy metals like cobalt and lead explain about 5 to 7.5% of variations, which they showed that the survey soils generally did not have a different overall communities between contaminated and non-contaminated sites. However, specific individual taxa were highly differentially abundant. As mentioned before, the study used gene markers, which are 16S RNA, 18S RNA, and ITS or FITS. The study does not focus on full functional metagenomic analysis. However, the study mentioned microbial survival mechanisms that supports the theory, which are efflux pumps and transport, detoxification and transformation of metals, sequestrations and bioabsorptions, and extracellular secretions.
The study has several strengths. It used three genetic markers and multiple soil depths, which gave a more complete view of microbial communities. It also used several statistical methods, making the analysis more reliable. However, some environmental factors such as pH and moisture and nutrient were not measured. In addition, fungal sequencing data was limited and some microbial groups could not be clearly identified. Many eukaryotic sequences could not be classified clearly. For the future improvement, include more soil parameters such as pH, moisture, and nutrients. Contaminants should also be measured at all depths. In addition, metagenomic analysis could provide more detailed information about microbial function and their potential for bioremediation.